Okay, so now, okay, for inhibitor that act on the allosteric sites, totally different, okay, from the inhibitor that act on the active sites, okay, totally different. Okay, so how they actually act on this? Basically, they bind to the allosteric site and then they interact with the allosteric site. So in these kinds of inhibitions, we call them as a non-competitive inhibition, where though the inhibitor going to bind temporarily or permanently to the allosteric site, because we do know that substrate won't compete with the inhibitor for the allosteric site, or vice versa. Inhibitor in this case, it does not compete with the substrate for the active site. Okay, so understand the concept here where they bind. This inhibitor bind to allosteric site, so they can bind temporary or permanently, regardless temporary or permanently down the inhibitor. Okay, this kind of inhibition is called as a non-competitive inhibition. So as long as non-competitive inhibitions, okay, we won't be able to reverse the conditions or reverse the inhibition by increasing the concentrations of the substrates. Are you clear? So increasing the concentration of substrate does not reverse the inhibition, because in this case, this kind of inhibition always decrease the Vmax, but Km remains the same. Okay, so again, let us look at what we're going to look to expect, okay, when we have the Vmax and Km together. Okay, so now look at this. So now, because when the inhibitor that bind to the allosteric site, when they bind to allosteric sites, this kind of inhibitor is called as a non-competitive inhibitor. When it's non-competitive inhibitor, we do know that if you plot the graph, this is initial rate of reactions, and then this is the substrate concentration. So this is the graph that without, okay, without inhibitor. Okay, and then if we draw a line from these regions, then we have the Vmax, and half of the Vmax, then I'm going to get the Km value. Okay, so again, what is the definition of Km? Km is the substrate concentrations that allow the reactions to achieve half Vmax. Okay, now, next, if you look at this, again, okay, so if with the competitive inhibitor, then my graph won't achieve the original Vmax anymore. They have a new Vmax, which is lower. Okay, oh, sorry, I think that draw it another way around better. Yeah. Wait, sorry, let me draw it nice. Okay, this is the Km. Okay, so this one is with a non-competitive. So if non-competitive inhibitor, so if I draw a line, I will get a new Vmax. So this Vmax is with competitive, and then half of the Vmax now, then you can see that Km remain the same. Okay, Km remain the same. Okay, so always remember for non-competitive, okay, for non-competitive, then you can see that Vmax now lower, Km remain the same. No change. Okay, so yesterday we also talked about this already. Okay, but now a little bit hard for us to explain, because what in our syllabus here, when you talk about Vmax decrease, Km remain the same. This one only applied to the, be careful guys, non-competitive inhibitions that bind tightly either to the active sites or to the allosteric sites. Again, guys, students always confused because we have 4 different kinds of interactions, right? Inhibitor. So inhibitor can bind to the active site or bind to the allosteric site. Okay, please follow. Okay, so when bind the active site, it can bind it, okay, temporary, or I say briefly, or can bind tightly. Same goes to bind to allosteric site. It can bind briefly, it can bind tightly. So this is what we have looked at it yesterday. So when you bind briefly to the active site, we call it as a competitive. Clear enough? Okay, and then you bind to the active site tightly, we say it's non-competitive. Okay, this one also called as non-competitive, and this also called as non-competitive. Okay, as long as bind to the allosteric site, they are non-competitive inhibitions. Now, here we have to understand, they bind to active site briefly competitive, Vmax will remain the same, no change, but Km increase. Correct or not? Yesterday we looked at it. Okay. Now, when we talk about this non-competitive inhibitor, okay, this inhibitor, when you look at the Km and Vmax value, only refer to those bind tightly, because this one, Km and Vmax, we won't learn about this. It's slightly different. Are you clear? Okay. So if the even though inhibitor bind to allosteric site briefly, they are non-competitive inhibitions, but Vmax and Km in the enzyme kinetics, they are different. Okay, so therefore we don't learn about this. As long as we talk about the Vmax for the non-competitive inhibitor, Vmax will be lower, Km remain the same. Are you clear? Here also Vmax lower, Km remain the same. There is no change. Now, why no change in the Km? Why no change to Km? Because the inhibitor already bind to the enzyme, so means that whatever effect gone already, so no more inhibitor. So that's why Km in terms of affinity, they are remain the same, because now enzyme only need to bind to the substrate, no longer have inhibitor anymore. So this group will be slightly different. We don't want to confuse you. So this part don't. And when talk about Km or Vmax, we cannot explain. Are you clear? We cannot explain. Okay, but the thing here is for us to explain, then we focus into these 2 groups for non-competitive inhibitor and this group for the competitive inhibitor. Okay, so come back to here. Okay, understand the concept here. Why Vmax decrease? Because inhibitor now already bound to the some of the enzyme, so means that those enzyme cannot function already. So there is a decrease in the workable enzyme. So therefore Vmax decrease. But why Km remain the same? Because Km in this case refer to affinity. So means that if as long as my inhibitor already bind to my enzyme either active site or bind to the allosteric site tightly, then therefore there is nothing to do with this kind of enzyme. They can't do anything already. They totally lost already. This enzyme cannot function. So the remaining enzyme actually only focus for the substrates only. So that's why their Km remain the same. Understand the concept here. Okay. So if you understand the concept, then I can go to the next part of the topic. So what is the effects now? Look at this. What is the effect of this inhibitor? So why this inhibitor will cause the inhibitions in this enzyme? Basically, when they bind, okay, here refer here, when they bind to allosteric sites, it's going to change the shape of the enzyme. Therefore, the shape of active site also change. Are you clear? Okay. So basically, it's something like this. This is my enzyme. Okay, this active site. So substrate originally can bind, no problem. Okay, but okay. Okay, now next, if let's say there is an inhibitor bind, okay, this is my enzyme again. So when the inhibitor bind, let's say inhibitor bind to this allosteric sites, what will happen here is they will change their shape. Okay, so the shape may be changed to this way. So when it changed already, now my substrate no longer can bind. Okay, substrate no longer can bind. So this is how allosteric site inhibitor functions. Whether they bind briefly or they bind tightly, it doesn't matter. Are you clear? It bind briefly or bind tightly, substrate now no longer can actually bind to the active site because the shape of active site now change. Okay, so highlight. So the net effect of the inhibitor is to highlight the shape of the active site. Okay, now initially they change the shape of the enzyme and then result in the change in the shape of the active site. But in the exam, we focus here, shape of active site. So it means that when active site change already, can you see that? So substrate no longer can bind, therefore the enzyme activity is inhibited. Okay, so we have two. Okay, one is the reversible. Now, in this case, reversible basically means that the competitive inhibitions can be can be stopped in this case. Okay. Now, look at this. I have this substrate. So in one of the metabolic pathway, my substrate have to be converted to intermediate A, intermediate B, and then end product. So this is end product. So I need to cleave it. Okay, make it smaller and smaller. Can I see that? So each step, they require a specific enzyme. Can you see that? Each step require a specific enzyme. So what will happen here is when substrate bind to this, when substrate bind to the enzyme, so definitely no problem. The enzyme still can function and result in the productions of the end product. But now, if let's say I have more and more end product already, more and more end products, why? The thing here is I want to stop the reaction. So what I can do to stop the reaction here is the end product increase in the concentration means the end product have a chance to bind to the allosteric site, change the shape of the active site, substrate now no longer can bind. So this feedback inhibitions here basically tell the enzyme say, yes, we have a lot of high concentration already, so means that we do not need to continue to produce the product. So the reaction stop. But the thing here is it depends on the end product. If the end product concentration is high, so some end product bind, end product drop out, okay, withdraw, and then another new end product come in. So basically, the frequency of the binding of this end product, which act as an inhibitor, it can continuously to inhibit the enzyme. But now, if end product is used, or end product is excreted out from our body, so when end product excreted out from our body already, concentrations of the end product decreases. So when the end product concentration decreases, so the chances for end product to bind to the allosteric site lower now. Can you see that? Initially, maybe every second we have one to bind, but now maybe 1 minute only one bind and release. So therefore, enzyme can actually functions back. Are you clear? So during the inhibition, now this is very important. During the inhibitions, enzyme cannot function, but after the inhibitions, yes, enzyme can function anymore. This is how we call it as the end product inhibition. So end product inhibition basically means that they are going to inhibit the enzymes, okay, but they bind only briefly. So the enzyme still can function as long as this inhibitor detach from the allosteric site. Are you clear? And we do know that the end product and the substrate, they don't bind to the allosteric sites. So therefore, there is no competition. So therefore, this kind of inhibitor is called as a non-competitive inhibitors. Are you clear? Okay. Now we have another one where the inhibitor actually bind irreversibly. So it means that it form permanent bonds with the enzymes allosteric sites. So example, irreversible non-competitive inhibitor act on the allosteric site is a heavy metal poisoning. So for example, argentium, okay, mercury, okay, a silver, like basically silver, mercury, and lead, they can form permanent bonds with this SH group of the cysteine. So still remember, cysteine and cysteine, we form the disulfide, correct or not? So this heavy metal can break the bonds between two sulfur atom or break the disulfide bond. So when it break the disulfide bond, then it's going to cause the enzyme lose the precise three-dimensional shape, and then you will know that these substrate now no longer can bind to the active site. Are you clear? Okay. So if you combine all together, now you can plot a graph without inhibitor and with competitive and non-competitive inhibitor. So how we plot it? So first, look at this, enlarge it a bit. So with competitive, very simple. You can see that you're going to go up. and then reach the Vmax. So blue line indicates competitive. Okay, inhibitor. Non-competitive. See, non-competitive, actually you can draw in such a way that not necessarily like this. I don't confuse you guys. So it can be any shape, guys. It can be any shape. So just draw it. It can be any shape and then lower. Okay, so this one is the non-competitive. Okay, so you can draw this graph. Not necessarily higher or lower, not necessary. So in this case, it's just an example to let you guys see the effect. Done? Here. So a quick summary, guys, a quick summary. So first, we look at this competitive inhibitor. So again, start with Vmax. Vmax will remain the same. Km actually increases. Okay, Km actually increases. Okay, for competitive. Okay, so this one only applicable to those inhibitor bind to active site briefly or temporary. Okay, and then for non-competitive inhibitions, so Vmax now become lower and then Km remain the same. So this only applicable to the inhibitor either binds to active site or allosteric site tightly or permanently. Are you clear? Tightly or permanently. Clear? So means that the inhibitor bind to allosteric sites briefly, we won't take into the consideration. We don't study that. Okay. So with this, I have done for the inhibitor.